Hi
Hello!
there.
Welcome to Microbiology Journal Club. We nerd out big on all things small.
My name is Danny. In a previous life, I dropped out from my PhD in microbiology at the University of Chicago, where I was infecting skin egg with MRSA. Then I was a fact checker for pharmaceutical advertisers, and nowadays I'm a member and the president of Biotech Without Borders, a nonprofit based at NYC dedicated to the public with the tools of biotech.
My name is Faz. I have a PhD in microbiology, and I've mostly worked on bacteria, but I've also worked as a research integrity specialist, and I'm currently working for a scientific journal.
Every week we need to talk about microbiology, or every other week we need to talk about microbiology. And today we're doing our news episode where we do an overview of the coolest microbiology papers that we've seen during the last 2 weeks.
And we also try to do deep dive papers. So if there's a paper that you think you want to get us to look into it in a bit more depth, make sure to message us at MicroTWJC if something has caught your fancy.
Yep, you can follow along with the papers that we cover on either week in our shared Zotero library, linked in the doobly-doo below.
And we want to hear from you, so please use the comments or tweet us at MicroTWJC. So, Danny, what kind of papers do we have this week?
Yeah, my selections for this week include Prozac stressing out bacteria, causing them to uptake antimicrobial resistance DNA, time-lapse visualization of bacteriophage coevolution, making better bacterial boba, a new HPV infection model using mice and a mouse virus, and freakishly sensitive measurements of microbial plastic metabolism.
And we also have commensal bacteria stabbing their hosts in the back the moment they stop getting fed. We also learn about viroids, which are RNA with attitude. Not only that, we also find out what a virtual microbiome can teach us about the flaws of microbiome analysis. Then we also look at how ants protect themselves against fungi and how fungi use that to proliferate. All that and more. And I think for lastly, we're going to be talking about our first paper, which will be about COVID mortality. Yes, we always start off with a SARS-CoV-2 paper because that's been our tradition for a long time to get the bad news out of the way.
Yeah. I mean, I think the rate of publication has gone down. And I mean, some of the lists that I used to look through to find new COVID-19 papers, like, they're not even being maintained anymore. But, you know, the pandemic is still very much in action. It's just people have adapted their behavior to find some intermediate space, not to be in total lockdown all the time. And this week we have sort of a retrospective bad news, I suppose. Assessment of COVID-19 as the underlying cause of death among children and young people aged 0 to 19 years in the US.
Yeah, so I mean, this is like a paper I thought to highlight because, yeah, COVID is still a thing, and it's important for us to keep track of that.
Mm-hmm.
And this one particularly stood out to me because of, well, because it's still like such a big cause of mortality, and people aren't talking about it anymore. Because a tragedy that happens once is a tragedy. A tragedy that happens every single day is just life.
Yeah.
I think COVID has now moved into that aspect of it's just life. We have to accept that the human lifespan is permanently affected by COVID-19.
Mm-hmm.
And so this paper looks back on August 21 to July 31st, 2022. So this is a year-long look that covers essentially the Delta outbreak and the rise of the Omicron strain.
Mm-hmm. Mm-hmm. Yeah, they pull that the death statistics from that period, and then they also have this list of like 113 selected causes of death from the National NCHS. What does that stand for? I don't have
National Center for Health Statistics.
Yes. Yeah, so they have this huge list, and they're essentially trying to figure out, given all of these these causes of death, where does COVID-19 in this study period fit into that listing? And yeah, they show, well, actually, I didn't. This was news to me. Like this U-shaped distribution of death amongst young people. I guess it makes sense. I didn't really think I hadn't thought about it before seeing this particular paper, but I guess the idea is that under 1 year is one of the bins in the 0 to 19 range, and then 15 to 19, and those are typically with causes of death. Those are the ones that are the highest, and then there's kind of a lull in between 1 to 14 years, which I guess it makes sense as like survival issues under 1 year, right? There's lots of like congenital defects happen at that age, right? A lot of like sort of serious genetic diseases are in that range, and so I think that's the reason. I don't really know why there's also supposed to be a or a jump at 15 to 19, but the authors say that that's like a very typical pattern for causes of death.
Yeah, this is interesting because I think another thing that stood out to me is there was a lot of talk during the outbreak about how children tend to be more resilient to COVID than adults and other populations. So taking that into account, I think this paper wants to balance that by making up the point that kids, the mortality rate of children is already quite low, that we're quite good at keeping kids alive from the ages of 0 to 18, and then after that they're kind of on their own. And yeah, this was interesting for me because especially this U-shaped thing, the U-shaped curve, where you see that most of the COVID-19 deaths tend to be in children under 1 year's age, the age group where it's very hard to get them vaccinated in time.
Yeah, to make a big difference. And I think that, but another element which they have in the figure or the second part of this figure is that, like, you can see that death also clusters around the Delta wave and the Omicron wave, right? Like July 2021 and January 2022. And those were, if folks remember from, like, living through it, like, those were things that, those were periods of the pandemic where, vaccines were very new and people were like, everything is back to normal. And like, everyone went back to these old behaviors, and then the huge spikes came. So it's one of those things where, and we've said it before on the episode, like, even if there is a really, like, low percentage of kids that end up having serious infection and bad outcomes, if you have tons of infection during certain times, that's just going to rise the levels of death and harm during those periods.
Yeah, I think that, like, so they basically took these records that have been collected about the COVID-19 outbreak, and obviously these records aren't going to be perfect, because of that some COVID cases aren't being reported as such, and vice versa. But and also they were going by these, like, clinical classifications, and they also filtered out any cases where COVID was just one of the causes of death. So if there's other causes of death that were attributed, those wouldn't count towards this. So these actually might be an underestimate of COVID-related deaths, because there is some controversy over whether, like, say, a death that happens immediately after COVID could potentially have been caused by it. So in order to get that sort of controversy out there, they were looking at deaths where the singular cause was SARS-CoV-2. And they ranked them with the other causes of death, and it's so it's pretty high up there.
Eight. Yeah, it ranked 8 in this time. And the stuff that's above it, I mean, actually, it's, like, also just horrifying reading this list of deaths, right? Like, stuff above it, like, there is, like, homicide is, like, actually a very high cause of death, like, within children and young people. Accidents, which I think is very understandable. Perinatal period. So stuff like very early. That's the number one really cause of death. Stuff like that's maybe very outside of medicine's ability to control. Congenital stuff is also above. And some cancers and heart disease also rank above COVID-19. But COVID-19, if you look at the all of the respiratory illnesses, then COVID-19 is the top of that list.
And of vaccine preventable diseases, it tops the list as well. So
influenza, influenza's there as well.
So it beats influenza, it beats measles, it beats varicella, it beats, like, hepatitis A. There's a lot of diseases that COVID-19 is topping things out of. But I think this is just to highlight that even in a situation where vaccines are readily available, COVID-19 is still something that we need to be careful about.
I mean, it's like almost, it says to me, maybe it's also like, what are the management strategies to deal with this? Like, maybe that's something that has to be important, like, given that it seems like it can't control, or like there's no will to control the COVID-19 rates around the countries, then maybe there have to be other safeguards put into place around places that are caring for children and young people, like especially in these high-risk populations. That might be like one recommendation that comes from this paper. And then I just want to mention one more thing about this paper is that they did say that this study period includes the Delta and Omicron waves, but they also looked at some data from like the beginning of COVID until Delta. And in that ranking, it ranked number 9 as a leading cause of death. So,
yeah. I think another thing that they don't talk about, which I think would be quite important, is the geographical distribution of these deaths, because different states behave very differently and treat COVID-19 in different ways.
Right.
And I think it might be useful to pass out whether there is the way those public health the way the public health response has been in different areas, whether that has an impact on child mortality or not.
Yeah, totally.
Might be.
I mean, definitely from the policy perspective, right? Like this is like a very zoomed out thing that like maybe it's not 100% applicable to where the actions and the policies are actually set. So yeah, breaking things out probably would help from the policy perspective.
Yeah.
Cool.
Yep. Moving on from COVID-19, we have a next paper, which is
antidepressants. Oh, go ahead.
After you.
Antidepressants promote the spread of extracellular antibiotic resistance genes via transformation. And I think this is there's a series of papers that have come out in the last few weeks about antidepressants and antimicrobial resistance. And one of them was behind a paywall on PNAS, so I wasn't able to, that wasn't one that we were going to share with you guys, but this one was open access. And specifically the phenotype that it's looking for here is transformation. So it's not really looking into what I'll say real world sort of scenarios, right? This is like a model system where they have reason to suspect that antidepressants might be increasing antimicrobial resistance. So here they go in and they model this, hypothetical scenario where bacteria in nature encounter some antidepressants because it's not a molecule that gets broken down in our body 100%. So people pee it out. If you're on it, you're peeing it out into the water stream, and it's sort of low-level contamination all over the place. And they see that when bacteria encounter these, they're more likely to take up plasmid. So they have a test plasmid that has some antimicrobial resistance on it, and they can see an increase in transformation efficiency up to a point as you increase the concentration of certain antidepressants. And we mentioned Prozac in the title or the teasers for this particular paper, and so that's one of them. I was looking up the brand names of some of the other stuff because I don't really know, and I don't really, none of them really rang a bell to me, but maybe it will for other people. Valdoxan, Cipralex, Cymbalta. I mean, they have different names also. Sometimes they're different. Lustral, and yeah. So Wellbutrin. These are some of the ones they tested, but 2 of them didn't have any effects they showed, but then 4 of them did show this increase in transformation efficiency.
Yeah, and they do delve into the reasons for that. So things like looking at the bacterial stress response, because I think there have been studies before that find that transformation is affected by when bacteria in stressful situations. They like to suck up whatever DNA is around to see whether they got any tools that can help them survive. Because
yeah, which is in some ways it's kind of interesting, right? It's nothing. It's not like these antidepressants have some magic capabilities. It's if they cause some stress, and I guess they don't all cause stress, but that's a really critical element. It's a very generalized response that they're inducing in these bacteria. And increase in competence is a very generalized stress response that happens. But I think they're able to, they isolate it to that stress as well. They perform things in an anaerobic condition, or they perform things where they have a oxygen free radical scavenger in place. And in those conditions, they don't see the increased transformation efficiency. So it's they have a sort of a stronger link towards this idea that it's a causal effect, right? Stress is caused by these antidepressants in bacteria, and then that in turn causes this increase in transformation efficiency.
Yeah, and like they delve in deeper with like the SOS response, like the like how certain genes are upregulated as a result of this, and like they produce more poor proteins that allow for transformation to occur. So I think this is
yeah. I mean, it's adding to that sort of core piece of knowledge that like has been that's being studied a lot in microbiology, and also like, I mean, for people that start doing like some microbiology or genetic engineering stuff right at the bench, you may wonder like, why do bacteria even want to take up DNA from their environment? Like, it seems like a weird strategy, but it's actually a very widespread strategy. There's huge amounts of the genome that are dedicated to the mechanisms to enable this particular action, and so it's interesting to see when they interplay in the environments, right? Like where these strategies have benefit for the bacteria. And yeah, this is one of those cases.
Yeah, I feel like the situation of when a bacteria finds itself in a new environment that's very hostile, it has this assumption that whatever's living here already must be adapted to it. So let's suck up whatever DNA is around, and then we might, by luck, pick up something that'll help us survive the way that the whatever already survives.
Yeah. I think that's a really like neat way of explaining potentially the strategy at play. So of course, places where this might be very important, like why this is important to know is because, like as I said, antidepressants don't get cleaned very well out of our wastewater treatment facilities, and so this might be a rationale to say like we should be doing better at that particular aspect. And then I think the authors also try to draw attention to the fact that just a lot of people are taking antidepressants, and so that's like a common thing. Like if you go to the hospital and you're taking antidepressant, like I guess it might put you at a higher risk potentially. I mean, they don't collect any of that data at all, but like the assumption would be like because this mechanism is in place, maybe you'd be at higher risk in some ways of like catching an antimicrobial resistance. I mean, there's no easy solutions, I think, like played out by this particular paper, but it's more just like drawing more attention and giving us like a mechanistic understanding of why this would be the case, why antidepressants might cause more antimicrobial resistance.
Yeah, I think it's interesting because antidepressants aren't the only chemical that we produce that will stress out bacteria. And so if this is something that's happening uniformly, then it doesn't just affect antibiotic resistance. I think it also affects like bacterial evolution, just full stop, because you're causing bacteria to become more competent and to take up more genes in order to adapt to the human lifestyle that we're kind of imposing on the world.
Yeah, I mean, in some ways you can see it as it means that like bacteria have all the tools, or they're in this mode where they are living in our built environments and they are actively adapting, right? Like evolution's being switched on overdrive for them to be able to find their niches within these built environments that we're creating.
Yeah, I think that's everything I wanted to say about this paper. But yeah, okay. Let's move on
to a novel lineage tracing mouse model for studying early MMUPV1 infections. So this one, when I teased this paper, I said HPV because that's the virus that's related to MMUPV, so human papillomavirus. I think people are familiar with. There's a vaccine for this particular virus. It has high associations with cervical cancer and as well as some other sorts of cancer. So MMUPV is the mouse version of that particular virus. And so these are DNA-based viruses that infect the epithelial layer, so the skin layer. And specifically, there's a lot of asymptomatic spread of these types of viruses, and there's not a lot of good infectious models, because infection models, you often have like a really clear endpoint that you're looking at. So like could be death, could be like a lesion size, something like that. I think it's been difficult to have that for HPV. But the discovery of this MMUPV, which is the mouse analog, and having mice as such a robust infectious model to begin with, has opened up a space to make a new infectious disease model to maybe understand this idea of like non-subclinical infection of HPV. And the way they do it is kind of fascinating because they want to make sure that they get their virus into one specific cell type so they can track it over time. So mice have this really great tool that's been in place for a really long time called CRE-LOX, where essentially you have specific mice that every cell, different cells have different promoters that are active, and so lots of scientists have done lots of work to have like a whole lineage of mice where they express this Cre protein only in specific cells based on what we understand of promoters inside of the genome. And so they're choosing a specific Cre mouse that has, I think, expressing it within their skin cells. And so then they're able to like add in this artificial plasmid that has all of the viral genes on it that they only get activated when the Cre acts on the plasmid and recombines it into the genome. And in that way, they're able to direct the delivery, essentially, of this virus only to specifically genetically identified cells within they're using mouse tail in this particular model.
Yeah, it's interesting because I think for papillomaviruses, they are essentially like they cause cells to proliferate. They cause them to have cancer. I think the interesting thing about this model is they create these synthetic DNA strands that encode the entire virus and then have these like lock systems with like a reporter gene. So when this actually enters a cell, it will integrate with them, and that will be like a permanent part of the cell during the infection. So when the cell starts to expand as a result of the virus, you can see that proliferation in real time.
Yeah, and because it's like, I think like the real strength is tying it to this Cre lock system, because it means that like you're not spraying virus and getting it into all the different cell types. Like it's a very strict genetic restriction where the virus is only replicating, at first at least, in this very specific cell type. And they put it in the basal layer because if folks are familiar, like skin kind of grows from the bottom out, and like we're constantly shedding our skin, and that's part of the defense mechanism. And so that's the cell type where that basal layer is the cell type where all other skin cells come from. And so essentially what they're able to do then in this model is track from the very beginning of this viral infection how that virus is changing, how the cells are being changed by that viral infection as it proliferates up through the layers of the skin.
Yeah, it's fascinating. So you essentially you're seeing the infection in real time. So there are a lot of studies that will tag the viruses, but I find it's really interesting that they decide to tag the cells that have been impacted by the viruses and that lineage. I think that's a very important part that might get neglected in some even in some thinking about viruses.
Yeah, I mean it makes things cleaner in some ways. Actually, the funny thing is it reminds me a lot of when I was working in like brain development, like a brain development lab. Is that like people studying brain development, like when things have a specific stratified layer, everything is developing from some basal proliferative layer, and by putting tags in that layer, you're following like the history, right? Because there's all this developmental programs that's happening, and the virus is interacting with that developmental program. So instead of having this average where everything got infected, everything's kind of like developing at different time scales, if you will. This method is setting that time zero as one cell type with virus, and then what happens at later time points, it's all synchronized in a way that they wouldn't be able to do if they were just using whole virus. And so yeah, they're able to show like a bunch of different antigens. There's a lot of genes that HPV viruses make, or papillomaviruses rather, DNA papillomaviruses make. So they're able to show like a bunch of them. As well, they even look at some TEM. So they definitely know that there's viral particles being made inside of these cells.
Yeah. So yeah, really interesting way of looking at this virus. So shall we move on to the next one?
Absolutely.
This is a really interesting one. I'll go. Multistep diversification in spatiotemporal bacterial-phage coevolution.
Yeah, I think this is a cool one. I mean, it definitely has cool videos that are associated with it. But essentially, they're talking about a model system, and we've talked about it before in the past, where you have a bunch of bacteria and you infect them with a phage, and then the bacteria evolve so that they avoid infection by the phage. But then the phage evolve to kind of avoid those defense mechanisms, and then there's like some equilibrium is reached. But the authors say that seems really odd that there'd be like such a quick, stable equilibrium reached, if you think about nature, like there's all these different variances. So the hypothesis is that inside of a well-mixed liquid culture, you don't get the same sort of like regional variation that could cause like different lineages of adapting phage and bacteria to grow. And so they moved this model into sort of like a semi-solid medium. So for looking at swimming bacteria, they often use like a softer agar, and then the bacteria that are motile are able to swim through that semi-soft agar. And so that's what they're using here. They inoculate with some bacteria, they inoculate with some phage, and because there's now like spatial separation, they actually see multiple waves of sort of like infection adaptation rounds. And of course, like those things are spread across the whole plate. So when they say they see multiple rounds, that's like by choosing like a small square within that whole plate, they can see like what the dynamics are. And yeah, and Fuzz, you're putting up some videos right now of like essentially what these experiments look like as they progress.
Yeah, you see that. So if we have a situation where everything, so here you see multiple waves as bacteria become resistant to phages, and then vice versa, where the phages again learn how to get over that resistance. And you see these plates that have all these all came from like the same like strain of bacteria, but then throughout this battle between the phages, you see all these different resistant colonies. And the thing that they were really going for here was to try and get diversity of resistance, figure out like all the different strategies that could be going through. And
yeah, because inside of that well mixed liquid culture, it kind of all goes to one. Like there ends up being like one winner. But here with the spatial separation, what they were able to see is that in different sections there could be different winners, and that's because like, you know, phage can't just like diffuse instantly from one side to the other. So it sort of matters this like structural arrangement in order to drive evolution, right? Evolution happens within an environment. So if you're creating essentially a plate that has multiple environments at play, you're going to see dynamics that you don't typically see in some well-mixed environment.
Yeah, you get lots of heterogeneity, and based off of that, they can sequence these areas of the plate and find out what strategies are happening in different regions.
Yeah, I think they end up doing maybe 2 major experiments, like one where they let it go once, and then one where they take the best performing phage and best performing bacteria of a previous plate and then seed it into the next set of plates. And they kind of like let that go on for a continuous cycle for some amount of time. And by the end of that, they have like, you know, there's tons of bacteria and tons of phage all over these plates. They can sequence them all. They do, I think, pairwise competition experiments between everything that they collected in their previous in these previous runs. And yeah, and they're able to create a huge matrix. And because they have the whole genome sequences out of that, they can then also pinpoint, oh, here's the genetic mutation that causes this particular strain to be better at infecting these bacteria, but not these bacteria, right? And vice versa. Here's a particular mutation that causes this bacteria to be resistant to this phage, but not this other phage.
Yeah, it's truly fascinating. And you know, I'm a sucker for videos as well.
Yeah, and this video too, it reminds me of there was like a really famous I remember like viral microbiology video where they had like increasing levels of antibiotic concentration, and then like the breakthrough when a single bacteria would become resistant to that and it would propagate through the plate. Like there's some it's reminiscent of those types of videos, I would say.
Yeah. It's essentially the same concept, except they're being resistant to the phage. And then, but the difference with it is that antibiotics can't become resistant to the resistance.
Yeah.
And that's why we can see these incredible patterns. Because what we're essentially seeing are battles between 2 organisms, and where the phages are kind of occupying the dark areas and the bacteria in the light areas. And so each of these plates, we're seeing almost like a battle in real time between the bacteria trying to spread out into the phage. And
Absolutely.
then you'll see, yeah, it is like mesmerizing once you like figure out what's going on. You can see like different lobes where different bacteria like cut off, and you see them each on their funny little journeys. And then occasionally you see like blankets of like blackness appear where the phages are winning. And then even in that, you can see little spots of slow-growing bacteria who are finding a way to survive even in those conditions. And then you have waves where a bacteria finds yeah, so it's really cool to see these in real time and to kind of have that idea that this is what's going on, and having that extra
I can imagine like just setting up like a bunch of these plates. You almost could create like an infinite stream of like sports commentary over the geography of the plate, right? Just saying like who's winning here and who's winning there. And of course, like you can't really say anything more because we don't have eyes that can see through the genetics of things. But like underpinning all of that, like there's all this genetic back and forth that's going on, and that's what the authors pull out of this paper, and they end up finding sort of certain genetic mutations that they hadn't seen before in other experimental models, causing phage resistance or sort of increased infectivity. And it's nice to be able to recapitulate things inside of a lab. So like, all of this is, nature knows all of this stuff, right? Like it's doing, these are all interactions that happen out there in nature, but having them inside of the lab in a way that they are spontaneously where they're occurring, right, based on the experimental conditions, like that allows us to go in and perturb them in a way that we can maybe understand why these mutations are important or what systems do these mutations play a part in, and thus understand the biology of phage and bacteria better.
Yeah, I mean, I'd watch that TV show. I don't know about you, but I'd watch the hell out of a TV show of that. Like Bacterial-Phage battle.
Just set up experiments and stream the commentary.
Coming to ESPN near you.
Yeah, ESPN 5.
Yeah.
All right, moving on. This paper we have, which is hilarious because I saw it shared like after I chose it to go onto the docket. I saw it shared on the Fediverse as well. So everyone's paying, we all pay attention to a common body of literature. This one's called A Stable Isotope Assay with 13C labeled polyethylene to investigate plastic mineralization mediated by Rhodococcus ruber.
Yeah. So this is an interesting, like, in-depth look at how bacteria can break down plastics, because I think there is this thing where plastics get washed into the ocean, and researchers just feel like they're more. We know there are lots of plastics in the ocean, but we don't necessarily know how they're getting degraded. And the question is, what role bacteria have in that degradation, and how we can actually measure it and confirm that it's happening, because there are other chemical processes out there that can cause it, like light can cause chemicals to break down.
Yeah.
But so we need to be able to distinguish that, and I think this paper is looking into techniques to fully characterize what bacteria are doing.
Yeah, I think that's a really good way to contextualize the paper. There are so many ways to measure whether plastic is disappearing. Like they even speak to like they can just like you can just weigh things with really sensitive scales, right, and see like what's gone missing, right? But this is one of the gold standard techniques that's used inside of sort of environmental metabolism, and that's 13C labeling. So if we say that bacteria are degrading this particular plastic, then what we're also saying is the bacteria is using the molecules inside of that plastic for its own body, right? Or releasing it into as carbon dioxide or something like that, right? Like if something's being metabolized, it's being broken down. And so C13 labeling, like most carbon is 14, right? Like that's the isotope of carbon that's around. But they're able to create a type of plastic that's enriched for a different type of carbon. And so then if you see this carbon released into the atmosphere of the bottles, essentially, is the experiment, or if you see these isotopes appear inside of the bacterial proteins or lipids, then you can sort of more confidently say, and also have this sort of very precise mathematical definition of how much carbon is exactly being eaten by these bacteria from this plastic that we doped in to the various experimental systems. So it's nice to see a very specific gold standard, if you will, set of data saying that some bacteria, I mean, you can see how limited it is, right? It's just one species of bacteria, this one type of plastic. But I think that the parameters that they calculate from this can be used inside of kind of more of the bigger ecological models talking about plastic degradation, that you can get like a better, more accurate estimation from the bigger theoretical models if you just like pass up the parameters from this small scale experiment.
Yeah, I agree. I don't really have much to add to that, because I think that it is, yeah, I think it is about precision for this, because I think we know this is happening, but being able to understand it is important.
Yeah, having the numbers for it, and then having the definitive proof that it's like the fact that they can show with stable isotope probing that it goes into the fatty acids of the bacteria that's growing. Like that's really good evidence.
Yeah.
And I think that that's something that, there's people are probably familiar with this news that like the mealworm, waxworm larvae, they can digest PET. They're like the gut microbiome of those organisms do so in some way. And so like I think that that's like it's important to remember that like that's one type of measurement, right? One type of measurement's gone into that. Like knowing, I'm not sure if the stable isotope experiments have been done in that scenario, and you could learn something if you applied it there. But it's not an easy technique to do, and like it takes time to set up these experiments and funding to get it done. So like we don't always have like the best data to support when something gets digested or not. And if you can get it, it can open up like a new vista. So like here, like as I said, they can estimate better in their theoretical assays. If they did it with the I'm sure maybe they have done it already. I'm speaking from a place of ignorance right now, but like, but if you were to see this matched data inside of gut microbiomes, you could even like pinpoint then the bacteria, right, that potentially are doing that metabolism, because you would see where the C13s are ending up. So like, I just want to point people's attention to this method, because it's like a very important method, a very difficult method to do, but get like some data from it that you can't get in any other way.
Yeah, I completely agree, and we'll pick up a little bit on the waxworm kind of thing a little bit later in another paper. So that's foreshadowing.
But for now, we're moving on to hierarchical encapsulation of bacteria in functional hydrogel beads for inter and intra-species communication. We've talked about this material before, hydrogel beads, sodium alginate. I think I introduced you to the term fruit caviar fuzz.
Yep, you did indeed. Yeah. I will never forget that.
Yeah, I think the previous time we saw this, it was like they were using like E. coli missile, which is a probiotic, and they were treating like mice with it in some way, and they were giving them like these like what I'm calling boba, because that's what they are. They're like soft jelly-like encapsulations of bacteria. But the reason the reason for this paper is they noticed that when they make these hydrogel encapsulated bacteria, the bacteria can spill out of it. And so they use their technique now like creates a shell around the initial encapsulated, the initial boba of bacteria. And the hope is that the shell will still allow things to diffuse across the barrier, but it won't allow the bacteria to get out. And then that means you can set up a whole bunch of interesting experiments that you wouldn't have been able to previously, including this idea of like nesting different shells of different bacteria, all sort of like within one unit. And Faz is showing us some of the videos that they've taken. Oh, and this is a video of one exploding.
Yeah, this is not what you want. You want, if you want to have bacteria in a shell, you want to keep it inside. But if it explodes, then that kind of ruins the point of having that shell in the first place.
Yeah, you can imagine, like, even without the shell, like, if the shell wasn't in place, then the bacteria could just diffuse outwards as well, potentially, right? Like, the ones that were, like, really close to the surface could swim away. And they have some images in the paper, like, showing, like, that particular phenomena. Yeah, and so this is an approach to study bacteria, and specifically they're thinking about how bacteria trapped within this hydrogel, that's kind of like a biofilm. Because that's like bacteria living in close proximity to each other inside of this matrix. And actually, alginate is a major component of the Pseudomonas aeruginosa biofilm. So, like, there's already this kind of like biomaterial compatibility, if you will, if we're comparing like nature to the model that we're creating. But they're using E. coli in this case just because they want something super tractable. And yeah, they're able to show, I think their sort of proof of concept experiment is they have this nisin production, which is just molecule that E. coli can produce. And they're able to stimulate the production of it. It's under the control of a specific reporter. They can stimulate the production of it even inside of the beads, meaning that stuff is diffusing into the beads and able to, activate different genetic circuits and so forth. Yeah. What else do they show? They show that that you can have different fluorescent bacteria, different layers of the balls. So essentially, like, you could have like a sort of a system of bacterial biofilms, multi-species bacterial biofilms communicating with each other within one of these balls. You could set up something where like the inside ball creates something in response to a signal that's on the outside shell. It's just a very flexible, there's just a lot of things that can be done here. And the fact that they're encapsulated just gives you the flexibility to say like the bacteria won't leak out in a lot of different circumstances.
Yeah, I think this is a great kind of, especially for like, say, if you have bacteria that doesn't like oxygen as much as another bacteria, you can be able to like stratify things. So maybe in the center of the bead, there's less oxygen around, so that bacteria will be able to survive, and outside they'll have more. There's a lot. I think another thing that this paper acknowledges is that bacteria live in a three-dimensional like environment, and we tend to forget that as microbiologists because we tend to work on petri dishes or even like homogenized kind of cell culture flasks. But the environment bacteria are used to are like complicated, like soil or other, and being able to acknowledge that, and also like multibacterial communities as well, where I think that's, I mean, especially for like cross-feeding and like methane, like kind of eating bacteria rely on other bacteria to. So I think experiments with these complicated structures for growing colonies are really interesting, and I feel like there is a lot of potential for the future for them.
Yeah, like in some ways you might be able to think of these boba as like zooming in to one spot in a biofilm, and where a biofilm it's going to like grow outwards and like it's going to develop and mature in some ways. The maturation inside of this boba is a little bit different, and maybe it could be like it's unclear what it models exactly. Like they didn't do that work to say like this is the genetic stuff that's happening inside of these boba that relate to the genetic stuff that's happening inside of these biofilms. But I could imagine that there is a very specific point in biofilm development that's being captured by these boba that they're creating, and that would mean that these boba are a really great model to study that sort of thing. But like that's not all, right? Like I feel like they also sell these boba as like a way of doing like biosensing, maybe. Like you could put your biosensors into these balls and you could roll these balls into different locations and then we'll roll them, I don't know, drop them into the liquid in different locations and they would like change color based on what was in that environment. So there's kind of like an interesting, the paper is a materials paper and it ends up like making me think of both applications within research sections, like sort of basic biology knowledge, but it also makes me think of like biotech things and like where might people use these, right, inside of some sort of industrial process or inside of like a diagnostic laboratory. Yeah, there's like a lot of stuff. Like as I said, like the last time we covered these boba, I think we were talking about delivery of probiotics and just like that, like this is a good way to deliver probiotics into some gut system. And so yeah, there's lots of spaces you could go. It's nice to see how sophisticated the research around alginate and bacterial materials has gotten.
Yeah, I feel like there is a lot of like potential here for like complications, and I mean, there's even like if you're working with a GM bacteria and you don't want to get in the environment, coat it with a bacteria that'll kill it the moment anything bursts or fades. I mean, things like that. That there are like all sorts of interesting things that we can think about that we wouldn't have the opportunity to think about with a homogeneous culture.
Yeah, absolutely. Oh, did you show them figure 5? I didn't see.
Figure 5. Let me see. Let me see figure 5 because
figure 5 is just like they're kind of it's very visual, but like they're just showing like how like they have different colored bacteria in different layers, and it just creates these like crazy iridescent orbs of fluorescent bacteria inside of the boba. Yeah, and you can imagine like, and I think this is I want to show it because like I think this is where you could imagine it as like a diagnostic thing. right? Like think about lateral flow tests and how like you look at bands that appear on like you know, this liquid diffusing up a piece of paper. Imagine, right? Like if you maybe you had like a little this little sensor that's floating inside of some liquid, right? And you like see that color change or something like that. Like it kind of made me imagine those things when I was looking at these pictures.
I'm just thinking about Tide Pods and how why do they have to make things look so tasty that you can
eat? Yeah, that's also very true. All right, let's move on to the next one.
Next one. Bacterial lifestyle switch in response to algal metabolites. So there are bacteria that live like in partnership with algae in certain situations. So phytoplankton can partner up with sulfites bacteria, and the thing is, the people noticed in this relationship is that sometimes the bacteria and the plankton, they can live in kind of harmony, and then every now and again the bacteria can decide to just kill the plankton host, and this paper is kind of analyzing the reasons why that is. So one of the things that they've been looking at is like what kind of signals do the algae give out that might signal to the bacteria that now's the time to start transitioning from being a commensal bacterium to being a pathogen. So this isn't just important for like algae, but it's also kind of important for all pathogenic bacteria, because there's quite a lot of pathogenic bacteria out there start off as commensals, and then when the situation arises, they can suddenly decide to go, hey, let's be pathogens. So things like
that is a very generalized understanding of opportunistic pathogens, right? And how do they work? They have to see some sort of signal. And I think what happens in this paper is they're sort of narrowing it down to a specific case of, as you say, phytoplankton and these sulfidobacters. A term that I hadn't heard of before, which probably makes a lot of sense now that I've heard it, is phycosphere. So like we've talked about maybe folks have heard of like rhizosphere, which is like around the roots of plants. There's like a zone where all the exudates from the plants are like influencing the microbes that grow in that area and influencing the the fungal colonies that grow in that area. Similarly, if you can imagine, even for a tiny unicellular algae, there's like a little zone around that algae that's the phycosphere, right? Or the area where molecules that those algae are secreting are being sensed by the bacteria that also live in that area. And so in some ways, I feel, in some ways, this model is supposed to be modeling the phycosphere of, but in a very one by one way. There's not like a whole microbiome they're looking at. They're only looking at like one bacteria.
Yeah, and one thing that they were focusing in on was the products that the algae might be producing to trigger this, because there is this they noticed that this kind of switch to pathogenicity comes when the algae enter into the stationary phase, where they've performed the maximum amount of algae that's possible within that situation. They start producing this compound called DMSP, and that seems to be an important like signal for the bacteria to go, oh, let's switch to pathogenicity. So they do a series of like transcriptomics experiments looking at like stationary cultures versus exponential cultures to see what other differences might be happening in the genomes. So what they're responding to look at the genes that might be causing that switch to pathogenicity. And they delve in, and they also like discover that there are things that like the presence of cause the bacteria to become pathogens. There are other things where the absence of it causes the bacteria to go crazy. So, for instance, there is a set of genes. There is a product. I need to scroll down to remember. Like benzoate. Yeah, benzoate is a key metabolite for the bacteria. So as long as the algae produce this algal benzoate, the bacteria are nice and fed. And they found that these bacteria can survive on algal benzoate. They really love that. And that if they give extra algal benzoate in the culture and add DMSP, a lot of the bacteria will decide to not go pathogenic, essentially. They'll still be commensal as long as they still get fed with benzoate. So,
yeah.
Essentially, this relationship feels like a bribe. And the moment the algae stop producing benzoate, the bacteria are like, yeah, right, we're done now.
Which is like, I mean, that's like, it's not I don't think it'd be incorrect to also think about like stuff in the rhizosphere as a bribe as well, right? Like plants can't move, right? So they have to like induce the behavior that they want to see, right, in the organisms around them.
Yeah.
The authors call the strategy of sulfidobacter switching into pathogenesis an eat and run strategy, because part of the pathogenic like upregulation is motility. So the thought is that in these giant, one of the theoretical implications might be that in these giant algal blooms, once they start starving a little bit and they're going to die, the bacteria that are associated with them are like, okay, time to now like destroy all these bacteria and disperse into the ocean. And so they'll turn on their ways of lysing everything, and then also their way of escaping into like a new part of the water column.
Yeah, exactly. It's ruthless, but
it's kind of ruthless.
And that's kind of what drew me to this paper, this story of like, because I'm always fascinated by when commensals turn out to be bad, and this is just one of those situations where you've got this tense, dramatic relationship between a bacteria where they're friends for as long as it's suitable for one of the parties, and then it goes Game of Thrones the moment that things get comfy.
Absolutely, but I think that this paper was setting us up to see that antagonism, because like we actually I'm not sure what the like, is there a benefit to having this bacteria around this algae, right? Like I feel like they didn't really explore like that side of it. Like, what's the bacteria doing for the algae in this particular case?
Yeah. I think they did mention that the bacteria were producing some vitamins or some other kind of nutrients for the algae to help it grow, but they really don't focus in on that in this paper. I think the focus here is on that switch of pathogenicity rather than the overall benefit of the mutualism.
because that's like, to me, that's like the sort of like the paradox sometimes inside of this question to in the switch to pathogenicity, right? Because if there is a state in which things are going okay, why would there then be these switches for when it doesn't go okay? And I think that like depending on how you cherry pick things out of the vast diversity of relationships that exist within nature, like you can kind of get different stories based on seeing different snapshots.
Yeah.
All right. Very cool.
Is mining metatranscriptomes reveals vast world of viroid-like circular RNAs. So viroids are very like interesting, like kind of. I didn't really know much about them, but essentially they're just like they are literally just RNA, and they find various unique ways to spread throughout, spread between different plants. So I think they tend to. They're mostly known from plants before this study. So they get spread out. They can get helped by accessory viruses, but essentially their main function is to find an RNA polymerase and trick it into transcribing its viroid RNA into more, and then just based on the structure, it can persist and it can spread like horizontally between plants, or sometimes vertically. And this paper essentially was going to a bunch of metatranscriptome like libraries and asking, have you seen these viroids?
Yes. And there's some characteristics that go into viroids that make them amenable to this type of metagenomic observation, right? Is that they have, a lot of them are with our RNA, and a lot of them have this hammerhead ribozyme going on, autocatalytic hammerhead ribozyme. They call it HHR in this paper. So like there are motifs that you can find. Also, a lot of them are circular closed RNA genomes. So like and I think that's part of why they don't get degraded easily, is because like something ligates them, so it's just like a loop of RNA. And so like using these two features, I guess, they're able to go mining. Yeah, they even call it mining. They're able to go sequence diving using algorithms and all this type of stuff into all the different metagenomic samples, which are essentially sequenced environments, right? They just took the free RNA and DNA within an environment and sequenced all of it. And they're able to find a bunch. It was actually, I did not know half the types of like definitions that they used in this particular paper, because they talk about like viroid-like RNAs, they talk about viroids, they talk about ribozyme. No, it wasn't that. It was some weird.
Yeah, there's a different new type of virus that they kind of talk about.
Ribozy-like viruses.
Yeah.
and like I'm not sure if people are familiar with this author, this like corresponding author, Eugene Koonin, but he's like he's considered like the originator of the RNA world hypothesis, where the idea of like what was life like at the very beginning, and could it have been a lot of like RNA molecules that just had ribozyme functions and did certain stuff like this. And I sense, so he thinks a lot about the definition of like what makes for an autonomous replicating object. And I think that a lot of his thinking is inside of this paper, because I can tell it's like the way that he's seen categories of stuff that might be considered life in some circumstances, right? How do we reconcile the diversity of that, or how do we find more diversity of this? Because that's helping flesh out maybe a thought of what the primordial living world might have looked like.
I mean, personally, for me, I'm thinking of this from like almost a biotechnological perspective of where we've got various like RNA like type treatments out there, the RNA vaccines, where there is a need to kind of expand RNA in certain cells to enact. And then we have these viruses that naturally do that in a certain situation. So what can we learn from that? There's so much diversity here, because there are like ribosiviruses, riboviruses, and yeah, I mean even like the Delta virus, which is like a helper virus for I can't remember, like hepatitis.
Hepatitis, yeah, hepatitis. It was hepatitis.
Yeah, and this is like an entire like world of, and I can understand why they might not have had this like kind of lens over them before, because if something infects plants mostly, it is something that doesn't tend to get looked at as much as say if it infected other life forms. So
totally.
Taking this out of that situation and then looking through these metatranscriptomes to find that actually they are very widespread in places that we wouldn't have normally thought about them being widespread can be very interesting. And there is like a massive like amount of information we can learn about them.
Sure. I mean, it just shows how diverse our world really is. Like we can find sort of new classes of self-replicating molecules at some frequency. I think they talked about like some of these RNA viruses or RNA viroids or whatever. They are parasites of transposons. So like other autonomous pieces of DNA, there could be parasites of that process. Essentially, anytime you're getting anything done in biology, there's the opportunity for errant pieces of information to be self-replicated along with the primary, right? Those primary self-replicating pieces.
I mean, that's incredible. Like, because we know that even viruses are viruses. Now we know that transposons can have viruses. It's just viruses all the way down.
But I just want to return to this. I mean, it's kind of a provocative thought to say like that there's like some biotech, like the way in which RNA vaccines come about, right? It's like somehow like harnessing the primordialness of RNA itself, right, to get stuff done within molecular biology within our organisms. I mean, viruses are the original hackers, right? Like of our biological systems. And I think like the stuff that we innovate, like, you know, I think ostensibly humans were also hacking the systems, right, to try to get better health outcomes for ourselves, right, a more healthy world or something like that. But like we don't, like we've been at it for a relatively short amount of time versus the sort of the long periods of time in which, just life itself has been able to innovate. So yeah, maybe, right, there's design principles that are happening at this level that we could somehow learn from. But I don't know what those are yet necessarily. I'm not sure if anyone is. Yeah, no, I don't know. I don't know.
Yeah, I agree. I think that I mean, the viruses haven't patented their strategies, so we're going to steal them.
We'll patent them instead.
Yeah.
All right, let's move on. We still got two more.
Yep. So
this next one is called the Virtual Microbiome, a computational framework to evaluate microbiome analysis.
Yeah, so this is an interesting one to kind of to follow on from that last paper, because this is looking at microbiome studies to looking at the way we study microbiomes, because I think that there is a lot of different strategies that people go out to to collect information about microbiomes, and this one focuses in on comparing DNA sequences, so looking at whole genome sequencing, I think metagenome sequencing as well, and amplicon sequencing, I think.
Yeah, like I think the interesting the information they're trying to pull out of these microbiome studies is differential abundance. Like when can you say that one set of microbes there's more of it than another?
Right.
And there's two ways that you could go about that. You could do the 16S, which is just like using PCR essentially to amplify this barcode-like sequence out from the organisms, or you could do you could just sequence everything and try to align those into genomes. We've seen a lot more of those papers recently because sequencing technology and computational resources have gone up. So now we get MAGs, right? Metagenome assembled genomes, where essentially they just like are able to do these very complicated algorithmic processes to pull out the full genomes of organisms living in a certain environment. And so there's these two techniques, and they can both tell us, if you use them on two different metagenomic samples, it could tell you what we think it tells us, right? Like sort of the lay person or like the lay scientist interacting with this data. They say, oh, it tells us that there are more of this one microbe in this environment than another. And what the authors are trying to show us in this paper is that there's a high rate of false negatives and false positives that can occur based on the underlying structure of the data that's going into these algorithms. And this is very reminiscent, actually. I put it in the linked papers. If folks are following along over Zotero, I linked a related paper to this. We'd spoken 2 weeks ago about a paper about microbiome differential abundance methods. So like almost a very similar angle to get at this problem, where it's like if you're using algorithms to try to figure this stuff out, you also sort of have to understand some of the assumptions those algorithms are making, and then and use that to cast a little more doubt on the certainness of the results that you're pulling out from those analyses.
Yeah, and essentially here they run the analyses that one would usually do on a microbiome, but using a virtual microbiome where they know what's in it, and they mix in a bunch of sequences, and they do different things. Because they understand that a lot of microbiome data is slightly incomplete, so they look at like how these different models are good at spotting, say, when maybe only small fragments of the genomes are available. And they look at the kind of how often they get false positives and false negatives. And the interesting thing is what they find is that even with like whole genome sequences, they find a high amount of false positive results coming through, even when all the information is available. And that it actually different. So like I think part of the thing is that when you have a large database with a lot of diversity of matches, it's a lot easier to find a match to anything. Which means that you're more likely to find a false positive match in those databases. Whereas with say 16S, that's a bit more constrained, but you're less likely to get false positives, but you're also less likely to get true positives as well. And and the interesting thing is like what I'd do in this situation. What I'd go is like, oh well, so we've got these two things that produce different outputs. Let's put them together and see if we can. So what if we added both 16S and whole genome sequences? Surely that would get more to a true result. And what they found here was actually no. Combining both of those also increases true positive, false positive. So this is a really like interesting paper if you're interested in looking at these kind of microbiome analyses. And
I will say that I think that the level that they're looking at is species. They're trying to talk about how many species are in these samples, but they find that when you move up a level and you're just trying to find out what genuses are in these samples, the analysis gets a lot better. Because I think the idea of going species, it's so specific that like, and maybe like the error rates or whatever at that level, like that drives part of this. But if you're willing to just say like, oh, I know the genus goes up, then like your analysis gets a lot more accurate.
Yeah, I think that is one of the big takeaways from this paper. And they also try to test this out IRL using a wax moth model. So wax moths we talked earlier, they can eat plastics, but the actual method by which they digest plastics is not quite well known. So there's a lot of microbiome studies trying to figure out what bacteria in their gut is doing the plastic munching. And so this study kind of is using that as a test model to to demonstrate what their thoughts about the microbiome predictions are. Like, and I think it's basically reinforces what they found from their virtual models.
Yeah. In some ways, it's like they made assumptions in making these virtual microbiomes. So like they actually then use an actual microbiome to say like, do we see the same patterns emerge using this actual data? Because they have to make assumptions. They made synthetic microbiomes that follow, the principles that they know of community composition. Yeah. So having an empirical test, I think, is quite helpful. And then also, like as you were saying, like it actually links to the idea that like what bacteria are doing this digestion, and I'll put the plug in. Like if people could do a really interesting stable isotope probing analysis of these systems, maybe we would be able to get to a better answer, right, than some of this. But yeah, that just goes to show the different ways to skin the cat in experimental research.
Yeah. So if you are doing 13C like studies on waxworms, I'm willing to offer the FAS prize, which is a full £5. That's all I have at the moment, but I will mail that to you for however.
All right. Let's move on to our last paper, which I found quite fascinating. Pathogen evasion of social immunity.
Yes. This is using a model of social immunity. So social immunity is like when a society cares for its sick. So it can be seen in, like, say, grooming practices among monkeys, or in this case, they look at ants, where ants are susceptible to funguses. I've seen a documentary known as The Last of Us, so I am now appropriately frightened of fungus. And ants, when they get infected with funguses, they look after their sick, so they will do certain, I think, grooming activities. So there are certain things that fungal pheromones that are given off that indicate that ant is sick, and the ants will go like, okay, let's groom them, cut off all the clean the spores off of them. And this model looks at what happens if you have an ant that's left all alone to get sick, and what happens if you have an ant but have some other ants there to take care of it, and see not only like what happens in survival, but also like how that affects different fungi species. So in these cases, they infect ants with lots of different species of fungi, and see which ones do well when only individuals present, and which ones do well when there's some social interaction going on, when they're being looked after by their friends.
Yeah, I think what's interesting is that in that social scenario, different fungi are more successful in that scenario than in the solitary scenario. And because this is an experimental model and they have control over all the variables, they can then take those fungal spores and they can do sequencing and find out what's going on. Like, why are certain fungus more successful than others?
Yeah, and what they found is quite interesting, because when like fungus is affecting an individual ant that isn't subject to treatment, the ones that like grow very fast and kind of kill the ant quickly tend to do better. But when it's in a social situation, it's the fungi that grow quite slow and don't show as many symptoms that tend to do better. So things that, like, they produce less ergosterol, which is that pheromone that clues ants in to say that, oh, this ant might be infected.
And actually, I think what's interesting about, so I said sequencing. They do sort of more of a, I think this is mass spec that they're doing in order to pick up the ergosterol signal. But what's interesting is that ergosterol, it's a known virulence factor. It's like a component of the fungal cell wall, and so it's interesting to see it have this linked behavior where it's linked to grooming behavior, but it's also linked to, like, more of it also causes more pathogenesis. So like it makes a lot of sense that ants groom when they see more of this molecule, because they know that molecule is something that is part of pathogenic. Well, they have some they have some knowledge, whether that's deep within their genetic circuits or like something that's passed down. That know that this molecule is associated with pathogenesis. So it's actually kind of fascinating to see that, like, that is encoded within the social behaviors of ants, and then you could also have scenarios where that it's being modified by the different types of the diversity of pathogens that actually do exist. Not all of them take the same strategy, of having a high amount of this particular compound.
Yeah, I mean, this reminds me of, I don't know if this is an old reference, but there's an old pandemic game where you're playing the bacteria and you can pick different strategies, and the best strategy is always to produce as much as possible so you can infect as many people as possible.
Right, it's like a flash game, and like it's like all the planes are the ones that transmit the disease around the world, and Madagascar doesn't have a lot of planes coming into it, so it's always the challenge to infect Madagascar. Oh god, we play the same video games. That's why we do this YouTube channel.
Yeah, that's right. We're a Let's Play, but now we're a Let's Read. But yeah, this kind of like the reason we doing a quiet strategy is it means that we don't alert the authorities to take control of it's the same kind of logic here, where the funguses that live can survive in the social situations are ones that don't raise those flags and are very stealthy. So yeah.
Just a very interesting, like, I loved it. Like, it's so simple in some ways, but it has all these like crazy implications, especially when you think of the social immunity as a model for like maybe other social behaviors that cause immunity. Like, this is grooming of ants, but like you can imagine it extending to like our own diagnostic and surveillance procedures, right? What does that mean for the diversity of organisms that get to cause disease in our society when we have specific types of diagnostic procedures? Like during the height of the SARS-CoV-2 outbreak, we talked a lot about S gene dropout, right? And like that fear that if we lost the ability to sense something, right? Like, and we saw in successive waves too, there was like it came back, oh, it went away, right? And there's an interplay between our diagnostic systems and evolution itself. And I feel like this is a very interesting paper to show us a real life example inside of a model system that then we can, go crazy in terms of imagining. But I mean, I think it's also good for ant researchers. I think that just studying the system in of itself can teach us a lot about how ants approach sort of social immunity, right? And maybe there's like design lessons within the ant population that are somehow relevant to the way that we work.
Yeah, and I think this, like, the way that bacteria can adapt to a society that treats its like own sick, I feel it feels like calls back to the Prozac paper we were talking about earlier about how bacteria are evolving as a result of all these chemicals that we're stressing them out with, and I feel like this almost puts another cap on another perspective on like how bacteria are adapting to antibiotics, and because antibiotics actually help bacteria that have antibiotic resistance in a way that weren't so much helped before. So, yeah, things to think about.
Well, that's all the papers that we have for you this week. Actually, the next time we'll be back is going to be March 26th, correct, Fuzz?
Yeah, that's right. I'm going to be disappearing for a month or so. But aside from that,
you can still interact with us over Mastodon. I'll still be probably trying to do this one abstract a day thing. And as always, you can like send us your suggestions and so forth.
Yeah, that's excellent work. Yeah, pay attention on Mastodon. Follow us on the Fediverse. I believe that's right.
Yeah, absolutely.
Yeah. We want to remind everyone that while we're very enthusiastic about microbiology and somewhat qualified, it's possible we didn't get everything right. Science is about thinking critically and asking the right questions. So if you have any questions or corrections, please let us know in the comments.
I totally agree. As I mentioned, you can reach out to us at MicroJC or MicroTWJC if you're still using the verb site. We both believe that peer review is a process and that we can all participate in it. So we hope that you had a good time listening to us ramble on about microbiology today. We'll be back in about a month. If you think you have something to add or found something unclear, please let us know.
It's been a pleasure chatting with you, Danny.
Same here, Faz.
Right. See you in a month or so.
March 26th. Bye-bye.
March 26th. Bye.